Welcome everyone. Today I'll be solving question number 39 of the 2023 GATE Biotechnology paper. This question asks you to choose from the given options events that lead to inactivation of tumor suppressor genes in cancer cells. The four given options are gene amplification, promoter methylation, loss of heterozygosity, and histone acetylation. So before we move on, let's look at a few of the terminologies mentioned here. So what is a tumor suppressor gene? A tumor suppressor gene is a gene that will regulate a cell during cell division and replication, and mutations in which can lead to uncontrollable cell growth and therefore cancer. Let's also look at each option briefly so that we can understand and choose from them correctly. So the first option is gene amplification, which refers to an increase in the number of copies of a gene in the genome. Like you can see here, this one gene has amplified into 2 copies of it, right? So that is gene amplification. Promoter methylation, which is the second option, is when a methyl group is transferred to the C5 position, that is this position, of a cytosine to form a 5-methyl cytosine in the promoter region. Okay. It can regulate gene expression by recruiting proteins involved in gene repression or by inhibiting the binding of transcription factors, and this results in gene silencing. The next option, or the third option, is the loss of heterozygosity, in which in a somatic cell becomes homozygous instead of heterozygous because one of the 2 alleles get lost. So like you can see here, these are the 2 alleles, one given by each parent, and in the cell, one of them goes missing. So this is what loss of heterozygosity means. The fourth and final option is histone acetylation, which alters the chromatin structure by acetylating lysine residues in the N-terminal tail protruding from the histones, these, right, in the nucleosome. So this process alters the accessibility of the chromatin, and it allows DNA binding proteins like transcription factors to interact with the exposed sites like this. First it was tightly packed and now it's exposed, right? And this activates the gene transcription for these genes. So now that we know in brief about each option, we can choose the correct options, right? Which lead to inactivation of tumor suppressor gene. Okay, so from these 4 options, we can see that promoter methylation and loss of heterozygosity are the correct answers, because promoter methylation leads to gene silencing, and loss of heterozygosity is where you lose a functional copy of the tumor suppressing gene and are left with maybe a mutated copy, and that might lead to uncontrollable cell growth, right? So therefore option B and C are correct. Thank you so much for watching.